So my name is Sato. I'm the CSO of Twist Bioscience. I also oversee the Biopharma team, which uses all of Twist's amazing DNA products for antibody discovery and antibody optimization. And today I'm actually going to be rolling out a new talk of mine. I've got a lot of new cool slides to show you, so I hope you enjoy it. I'm going to talk to you today about both our library of libraries, which we use for antibody discovery, as well as our optimization platform that we call Tau. But before I do that, I really would like to kind of roll out all the stuff we're doing at AET. First of all, big shout out to our friends at Abveris, who we recently acquired, and they're part of the Twist family right now. They actually have multiple talks as well during this meeting, and you should all go and check out their talks after attending my talk, of course. Second, definitely go check out our booth. We're giving out a bunch of really cool llama giveaways. And then finally, we're also doing a big mixer on Wednesday. Our QR code for it's up on the screen if you can snap it with your phone, but also there's a little pamphlet in front of you where you can also scan and use the QR code to actually sign up for that event as well. And then finally, we also have 6 posters from Twist Biopharma. So we have a lot of scientists that have been wanting to come to a conference for a long time and actually present some of their good work that they did during COVID. And so please go check out their posters as well in the exhibition hall. So I'm often asked, why Twist Biopharma? Why should I work with you? And I'll get into more kind of technical details why we believe our approach is unique. But the first is that, as you may know, Twist is a DNA company, and so we take a completely synthetic approach for our library business that we couple into Twist Biopharma. And in our mind, that really helps us reduce the risk. We can, as you'll see later, we can remove a lot of things from our libraries, as well as put a lot of human content into our libraries to make sure that the final antibodies that come out are fully human and potentially devoid of any developability risk. In addition, we can discover fully human antibody leads very fast. All of our libraries are in phage display. We can pull them out of the freezer, and within a matter of weeks, we can have potential hits against your target. And then finally, we're extremely flexible. As you'll see about the end of my talk, there's all kinds of ways you can work with us, from licensing our libraries to having us do projects for you, to helping you develop and build a custom library. So there's all kinds of ways that Twist Biopharma can help you. So when you think of us on a macro level, in our mind, everybody's really racing to get to the finish line to find their particular biologic drug. And you should really think of Twist Biopharma as a division of Twist Bioscience that can really help you discover, optimize, and develop drugs really quickly using the platform that I'll talk about today. So those of you that are maybe familiar with Twist probably know us for a lot of our DNA products that we buy and that we sell to the open market. And that's really our synthetic biology business, where we, of course, make clonal genes, gene fragments, oligo pools, DNA libraries, so you can use us for all kinds of applications that involve DNA. We also have a whole NGS side of our business where we use oligo pools, basically pull down specific exomes ahead of NGS sequencing. And so that's a huge part of our revenue generating business. But a couple years ago, we actually kicked off a couple new divisions of the company. One, Biopharma, which is the group that I started up in the company, and then data storage, which is the idea about using DNA to actually do long-term data storage. So when I often talk about Twist, I often love to start with our core-based technology, because I always say that the best companies out there understand the one thing they're really good at. And for Twist, that's our ability to print DNA. So we have this amazing silicon platform where we can actually print up to 1 million individual oligos up to 300 base pairs in length. So see it as like an inkjet printer, but instead of colors, we're actually printing individual oligonucleotides and actually making long oligos on this silicon array. And so it's an amazing system for making oligo pools, and we use those oligo pools to make all the custom DNA products that I talked about before. And so Twist Biopharma is one of the few, probably the only, antibody discovery company that's actually built on top of a DNA company. So we have immediate access to clonal genes, libraries, all of the amazing products that I just talked about. And so in my mind, that really gives us a huge advantage in this space. First of all, because we're so good at putting together DNA from oligo pools, we can generate high quality libraries really fast, much faster than anyone else in the industry. And so as a result of that, today we actually have 18 different libraries in our library of libraries. So a lot of our competitors, they may have one, because building a library is just really hard. But because we have this amazing expertise in library construction as well as the silicon platform I just talked about, that allows us to be really innovative and create libraries that no one else can build in this industry. Second is quality. Even though we're using synthetic libraries, we're basically using oligo pools in the generation of all those libraries. And as a result, as you'll see in a second, we can actually put in fully human content in those oligo pools that we use to generate the libraries. So even though we're making a synthetic library, they're very human in their nature. And then finally, speed. Twist is an amazing place for automating every single part of the process of DNA synthesis. And actually, we used a lot of the learnings from our automation team at Twist to actually automate and speed up the process of antibody discovery as well. So when you look ahead to enriching your pipeline with antibody drugs, you should really see us as an instrument to help you in that endeavor. So we really love to focus on that we can, from our libraries, discover drug-like fully human antibodies quite readily through the process I'll talk about in a second. We focus in our design sets for all of our libraries based off known antibody sequences in different spaces. And so as you'll see later when I talk about each individual library, I'll talk a lot about the design set. What source of antibodies did we use in the initial design of the library? And in all cases, that design set came from a known set of antibodies from a particular space of sequences. And then finally, as I mentioned before, we have this huge broad base of libraries. As I said, we have 18 different libraries today. And so that gives us a huge breadth in different scaffolds and different diversity sets derived from those libraries. So in terms of the advantages of using oligo pools to generate libraries, I'd really love to show these as potential advantages of that approach. So when you think of synthetic libraries, you may think of people using degenerate oligos, either trim-based oligos or perhaps mixtures of nucleotides as they add in combinatorial diversity into their libraries. Here we can actually use oligo pools in the generation of those libraries, and I'll show you in a second how we do that. But that gives us amazing control as we build our libraries. We can actually exactly print the sequences that we want in all of our antibody CDR loops, and that gives us really tight control as we build our libraries. One advantage that I love to cite is that we can also very easily change the length of our CDRs. In particular, for example, heavy chain CDR3, which you know has a huge length diversity. We can actually just make different lengths of oligos as we build those libraries. And so that allows us to, in very short period of time, build a very natural library that has different lengths of oligos, also different diversities, all derived from natural human content. So Twist Biopharma, we're a division of Twist Bioscience, as I said before. We're very focused on helping others on the early stages of antibody discovery as well as antibody optimization. We're also using this technology for ourselves as well to help us generate our own pipeline of drugs derived from our libraries as well as to help optimize them. Down the road, we will eventually get into some of the later stages of development and clinical development, but for now, we're very focused on the front end of that discovery funnel. As I mentioned, our business is really segmented into two components. On the discovery side, as I mentioned, we have over 18 libraries in our library of libraries, ranging from Fab to single chain to VHH single domains, and they're all in phage displays. So they're easily, readily available to pull out of the freezer and at a moment's notice, start panning and screening against your individual target. And I'm gonna go into the specific design for each of these libraries in this presentation. And then second, on the optimization side, we can actually take any antibody lead derived from any platform, not just ours, and then we can actually use a database of natural human sequences, and from that actually design a very focused library around your specific lead. And if it's a mouse-derived antibody or a rat-derived antibody or even an antibody from a llama, we also can humanize it at the same time. So this is an amazing platform for affinity maturation where we can shuffle natural human diversity in the context of your particular lead. So in terms of the workflow that goes into what we do, again, as I said, we have these amazing libraries. They're sitting in the freezer. We basically take your target, which could be a recombinant protein. It could be a cell that overexpresses the target. It could be a synthetic peptide derived from the target. We can pan and screen it using these mixtures of libraries. Oftentimes what we'll do is we'll pool various libraries based on scaffold. So we'll have a Fab pool, single chain pool, and a VHH single domain pool. And we'll basically do usually 4 to 5 rounds of panning against the target. After doing multiple rounds, we then do screening, ELISA-based screening if it's a recombinant protein target, and pull out all of our ELISA positives. In addition, we often do for our cell-based targets as well as our recombinant protein targets, do full NGS analysis of every round. And that gives us a lot of information about which sequences are actually enriched through the multiple rounds of panning. And then finally, again, taking advantage of being inside of a large DNA company, we typically make 100, maybe even 200 antibodies directly from those results. We go directly into full-length IgG if that's the modality we're going after. We make and express them internally at small scale. And then we put those antibodies through a whole battery of tests, and that might be doing flow cytometry to look at cell binding, might be looking at affinity, as well as putting them through a whole battery of developability tests. And so within a matter of, let's say, a quarter, 8 to 12 weeks, we can actually, in very short order, come up with fully human leads against your particular target. In addition, initially within Biopharma, we were doing a lot of antibody expression and purification. And again, it was my dream that we would offer this up to the greater world, because a lot of our customers were already ordering a lot of antibody genes from us. And so as a result of that work with the SynBio side of the business, we're actually moving to kicking off a new product within Twist to not only offer the people the ability to synthesize antibody genes, but also there's going to be a dedicated team within Twist to actually make and purify and deliver small amounts of antibody from 100 micrograms even up to a milligram in scale. And we'll be doing that all in XB293 to really create and offer back to you the ability to make hundreds and thousands of antibodies for your screening efforts. And we believe that for folks that are doing a lot of, like, for example, single B cell screening, where they're oftentimes making synthetic genes, this service will be really instrumental in their workflow. So as I mentioned, our viewpoint around libraries is that one library is not enough. We really strive to have a library of libraries, and it's my dream someday to actually have a whole plate of libraries. So in phage display, we're oftentimes limited on our total diversity, which is usually around 10 billion in a library. And so if I actually have 100 libraries, I'll have 100 times 10 billion, or 10 to the 12th different antibodies in total. So, and those could range from different scaffolds, and the diversity sets could be derived from all kinds of different spaces of antibody sequences. Since the universe of sequences is literally almost limitless, if we focus it in different pockets within that allowed diversity, as well as provide different scaffolds, we can adequately sample the allowed space of antibody sequences. Okay, so I've talked a lot about, on a high level, what Twist Biopharma does. Let's now dive into how we actually build a library. So as I said before, we use alga pools as we generate our synthetic libraries. And so we can actually make 1,000, 10,000, 100,000, maybe even 1 million sequences that are derived from nature in each of the different regions of an antibody. So let's talk about a VHH single domain that's basically encodes 3 different CDR loops. We can actually make an alga pool for each of those different loops, and then basically splice them together in the context of a single human germline, or maybe even a llama-based germline framework. And we can basically shuffle those CDRs in different combinations to give us antibodies that bind any target we wish. And as you'll see later, we can actually use natural occurring CDRs as we do that and find unique specificities to any antigen that we go after. We also can, I often love to talk about what we can actually take out of the library. So even though sequences derived from animals are often devoid of any developability risk, there occasionally do creep in some sequences that might be prone to developability issues. So we can actually take out those sequences from the alga pools before we generate the library. And then finally, the use of alga pools also allows us to really cool things like insert specific motifs into our CDR sequences as well. And that's actually key and instrumental to our target class specific libraries that I'll talk about later. All right, so now I'll dive into the library of libraries. I'll spend hopefully about 20 minutes going through all of them. I'll then dive into tau, and then I'll finish off my talk. The first one I'll talk about is our GPCR 2.0 library. It's built off of this premise by this paper back in 2014, where they actually hypothesized, they took this known crystal structure of CXCR4 bound to this loop peptide. And what the authors hypothesized is, could they actually take this loop peptide, which is a known peptide antagonist of CXCR4, and actually graft it into a long heavy chain CDR3 of a bovine antibody. And what they were trying to do is they're trying to see whether they actually could create an antibody antagonist of the receptor rather than a peptide that would retain the same antagonist capacity as the original loop peptide. And that's exactly what they found. They found that the grafted antibody actually had the same antagonistic function of the loop peptide. And so what we wanted to do is take this a step further. Could we create a fully human antibody library, not a bovine one, that had a long extended heavy chain CDR3? But rather than just have one motif, what if we created a library of motifs on the order of hundreds and thousands of things that are known to bind to GPCRs and actually fold it into the heavy chain CDR3 of the library? And so that's exactly what we did. This library was actually initially built in collaboration with Distributed Bio, that is now part of Charles River. We worked with Distributed Bio to actually generate this library. And the first thing we did is really collect all those motifs that are known to bind to GPCRs. And those range from known peptides derived from chemokines that are known to bind to GPCRs, as well as helical peptides and loop peptides. A lot of GPCRs actually interact with themselves. Their N-terminal domain actually folds over and interacts with their second extracellular loop. So we took a lot of peptide content derived from the N-terminal domain. And then finally, even though finding antibodies to GPCRs is really hard, there are a fair number of them known. And so we actually folded in a lot of heavy chain CDR3 content from those GPCR antibodies that are known as well. And so after about a year and a half worth of work in collaboration with DBio, we actually were finally able to build the final library. It's made of multiple heavy chain scaffolds, as I'll show you in a second, as well as multiple light chain scaffolds. We did a lot of work to show which heavies and lights worked well together, as well as which long extended heavy chain CDR3s could display on phage. We did a lot of work on displayability, and when we felt that we had a set of scaffolds and a set of extended heavy chain CDR3s that could display very well, we then put the final motif library into it to actually build in this huge array of things that bind to GPCRs into heavy chain CDR3. And so this is actually the library design. I've actually broken it down into the actual individual oligo pools just to really show you how complex these libraries are. And it really gives you a flavor for how we build libraries in the sense that we basically create multiple oligo pools that encode for separate regions of the antibody library, and then we piece them all together in the context of multiple human germline frameworks. And the bulk of the library that was really the main driving force in the library was that heavy chain CDR3 loop, which had a huge array of different GPCR binding motifs. And as you can see, we had over 100,000 different GPCR binding motifs in that one region of the library. And so using this library, the proof of concept that I love to show is actually a proof of concept that we did against GLP-1 receptor. It's a class B GPCR. So we pan and screen the library against cells that overexpress this GPCR receptor. And we found a huge number of hits that bound with high affinity to GLP-1 receptor. When we actually went back and looked at the heavy chain CDR3s in those leads, we found that many of them actually had the GLP-1 peptide, which is the known ligand for GLP-1 receptor. One of these antibodies, TB01-3, was a very potent antagonist of the receptor, completely blocked signaling through a competitive manner, and also had long PK as well as activity in an efficacy model. We've now gone on and actually made a single domain version of the same library. In my mind, single domains are actually uniquely suited to bind to and potentially block GPCRs because they have a much smaller size compared to a heavy chain, light chain antibody. So we want to build in some of the same motif-based content into a single domain library, which is why we built a VHH library using that same heavy chain CDR3 motif content. One criticism we got from the GPCR 2.0 library is some people weren't crazy about putting motifs derived from non-antibody content into heavy chain CDR3. So instead, we actually built a GPCR library purely focused on antibodies that are known to bind to GPCRs, and that's the library that we call GPCR 3.0. And so from that, we actually built a library of 2 heavy chain as well as 2 light chain frameworks. We folded in over 61 patented GPCR antibodies that bind to 22 different targets and folded in all of the content for all the CDR loops from those 61 antibodies. And this is just showing you one example where we pan and screened against CXCR5 and found a very potent antagonist of CXCR5 that actually block the signaling in this cyclic AMP assay on the right. All right, now we'll get into our Hyperimmune library. Again, here's another library where I basically want to do something that no one else had ever done before. So I wanted to really take advantage of the silicon platform to create an oligo pool with over 2.5 million heavy chain CDR3s. So I combed the databases, collected as many heavy chain CDR3s as I possibly could find that were all unique, that were derived from both naïve as well as memory B cell content. And then I took over a synthesizer for a couple weeks and made over 2.5 million heavy chain CDR3s to put into that particular register of the antibody library. We also folded in antibody diversity into the other 5 loops of the library, and then we put it into a Fab-based scaffold that had VH3-23, VCAP of 1-39. We also eventually made a single chain version of the same diversity as well, so we can either use Fab or single chain and actually select and pan the same diversity. And then we finally transformed it again on that 10 billion level. And then finally, this slide actually goes through all the details in terms of the humanized framework that we use, as well as the overall diversity. As I mentioned, we really wanted to put a lot of diversity into heavy chain CDR3, since that's oftentimes the primary determinant in binding. And what we're finding when we use this library is that we oftentimes see that we get a wide range of different epitopes. And of course, the epitopes are basically clustered based on heavy chain CDR3 sequence. During the pandemic, we actually used this library to pan and screen against SARS-CoV-2 spike protein, and shown here is just an example where we pulled out 3 different antibodies that bound different sites on the spike protein. These particular antibodies were quite unique. They only bound the up conformation of the spike and not the down one. And they bounded a site that actually was very potent in terms of neutralization. Shown here is just some more data on those leads, where we did a cell-based assay looking at competition between spike and ACE2 on cells, and we see really potent nanomolar competition between our antibody and S1. All right, now I'll dive into our VHH single domain libraries. We actually have four libraries I'll talk about now, plus the 5th one I talked about before, which was our GPCR single domain library. Here, again, we start from a design set. Here, we actually started with over 3,000 VHHs derived from llamas that all had unique antigenic specificity, and that all of them bound a single antigen that was known. We used that design set to then create the 4 libraries I'll talk about now. The first one is our VHH ratio library. So we took a consensus llama framework derived from those 3,000 VHHs, and then we took all the amino acids in CDRs 1, 2, and 3, and we created mixtures of ratios of amino acids in oligo pools that kind of mimic the diversity seen in those 3,000 VHHs. So kind of like a degenerate oligo, but in a much more controlled way, where we would sample each of the amino acids at each of those positions as shown here on this slide. And so that gave us a huge amount of diversity, as well as we were able to couple in a lot of length diversity into the CDR3 register of the library as well. The second library is what we call our VHH shuffle library, which is still built off of a consensus llama framework. But here I wanted to do something a little different. Rather than use mixtures of oligos that mimic the ratios of amino acids that are allowed within the diversity set, what if I could just shuffle the natural llama CDRs as they exist in the database and come up with unique antibodies that bind any target we wish? And that's exactly what we see. And what's really cool about this library is the overall theoretical diversity is actually lower than the practical diversity. So that actually allows me to fully select and screen the entire diversity of the library, which is quite unique for a phage display library. The third library is what we call VHH shuffle. Same idea as the previous library, but now we're actually putting it onto a humanized trastuzumab-like framework. So the VHHs that come out of this library are actually very human and can be used for all kinds of other, can go directly into humans because they're already on a humanized scaffold. And then finally, the last library is, we actually took some learnings from the Hyperimmune library I talked about before, where we took the VHH shuffle library and we actually replaced the CDR3s, which again were originally derived from llamas, with the human heavy chain CDR3 derived from the Hyperimmune library. So here, this library actually has a humanized framework, and it has human heavy chain CDR3s coming to the CDR3 position of this VHH single domain library. And when we've gone on and used this library, they've been very robust. We've been able to get 50 plus hits from each of the different libraries against any target we go after. Shown here is just where we used it against TIGIT, which is an immuno-oncology target for those first 3 libraries. And as you can see, we got a huge number of uniques from the library. We see a lot of length variation in CDR3 based on library. And then when we actually look at the affinities of these VHHs in a monovalent setting, we're seeing affinities in the range of single digit to triple digit nanomolar. We also went on and used these same libraries for COVID as well, and we actually came up with a whole range of single-digit nanomolar, even some sub-nanomolar binders to the spike protein that were very potent in blocking and neutralizing SARS-CoV-2. And this is just showing you some additional data. Again, competition cell-based data where we're trying to look to see how well do our VHHs that bind the spike protein actually compete between spike ACE2 interaction on cells. Now I'll dive into our scFv libraries. The first one I'll talk about is our SAB library, our structural antibody library. Here again, our design set was all humanized antibodies that have actually had a crystal structure determined, with this idea that antibodies where they've been crystallized potentially are very stable, very developable. We took this as our input data set to create this particular library. And so shown here is the design for the library. Again, we started with a VH3-23 VKappa1-39 base scaffold, germline scaffold for the library. It's in an scFv framework. And then we're just shuffling the CDRs from all 6 loops derived from that initial design set of all the antibodies derived from that particular germline in the PEV. And we showed that we can get all kinds of high affinity binders to any target we wish from this library as well. Shown here is just one example where we actually pan and screened against CD3 and actually found human cyno cross-reactive clones to CD3, which, as you may know, is actually a really difficult thing to do from a library. The next library I'll talk about is what we call our Ancestral library. Here, our design set was actually antibodies derived from the patent literature. So we basically took all human antibodies that have ever been patented, and we took that as an input data set to again, shuffle the natural human diversity derived from those patented antibodies and find unique specificities to other antigens. So shown here is, again, the ALGA pools that constituted that data, each of those different antibodies from that database, and then we, again, folded in and put this into a VH3 VKappa1 germline framework, and then folded in all those CDR sequences derived from the patent literature. To do that was actually quite difficult because there's so many sequences in the patent database. So we actually went through a whole process of trying to narrow down our sequences into smaller clusters. And so we actually used a full informatics clustering analysis to find representative sequences within each of the different CDR loops so that we could limit the number of CDR sequences that we would put into the final antibody design. And then we finally showed, again, using the spike protein as a test case, we were able to get high affinity binders to the spike protein that also competed with spike ACE2 interaction. And then finally, the last library that I'll talk about is our scFv library I'll talk about is our AI hyper-mutated library. Here, we actually work with a company called Deep CDR, which what they did is they actually simulated over 1 million antibody sequences derived from known sequences in the human antibody database. We use those millions of sequences to actually design the 2 different heavy chain as well as 2 different light chain sub-libraries that are built into this library. We also showed that the final library that was built had very similar allowed amino acids that were similar to the original design set. And as a result, we believe it's a nice survey of sequences that are, again, fully human-derived. And similar to the previous libraries, we pan and screened the library against SARS-CoV-2 and found a whole range of sequences that bound with high affinity as well as blocked the spike ACE2 interaction. Is our Minotaur library. Here we work with another company called Taurus, which is actually a local San Diego company that was acquired by Ligand. Here, they actually focus on bovine-derived antibodies. And so kind of similar to the GPCR libraries I talked about before, but here we wanted to actually incorporate long heavy chain CDR3s from cows. We really focused on the long rather than the ultra long sequences in cows. And the idea was, cows have these long heavy chain CDR3s for a reason. Perhaps, we could fold those into a humanized scaffold and really take advantage of some of that content that the cows have, through evolution, created. So again, we created a fully human library. We put human CDRs in all of the other CDRs except for heavy chain CDR3, and we incorporated the long CDRs derived from the cow into the framework. As you may know, cows actually have a lot of cysteines in their CDR3s, as well as sometimes disulfides to the framework. And so we actually segmented all of those sequences into different populations based on their cysteine content. We created a library with no cysteines in CDR3, one that had even cysteines where there was actually a disulfide in the CDR3. And then finally, we also created an odd cysteine library where there's actually a disulfide with the framework that many cow antibodies also have. And so from that, again, we were able to find human cyno cross-reactive clones to CD3, really showing the power of this library to find very unique epitopes on targets. All right, finally, I'll end with a couple libraries where I don't have any data, but they're, again, new libraries we've created, and we're looking for customers that we'd really like working with some of these unique libraries. The first one is what we call our ion channel library. We took a similar approach as the GPCR library where we incorporated motifs, but here we actually incorporated motifs on things that are known to bind to ion channels, which, as you may know, are derived from like venoms, derived from various organisms, as well as all kinds of other really nasty peptides. We actually incorporated them into these antibodies because they're, again, known to bind to ion channels and actually elicit a very potent function. And so as a result, we were able to build a highly diverse library which had a huge array of different motifs that are known to bind to ion channels. The problem with working with ion channels, though, is it's oftentimes really hard to actually get the target. So we've been having a hard time finding a customer that actually has a cell line that overexpresses their particular ion channel to actually test out this library. So if you actually have access to a particular ion channel you want to find an antibody to, please follow up with us, and we'd love to do this as a test case to really understand the power of this library. And finally, this slide here just kind of shows you how it was designed in the sense that we use both cysteine-containing peptides for CDR3 as well as no cysteine peptides, and we created these two different sub-libraries within the ion channel library. And then finally, another library where we don't have any data, but we're again looking for partners that are willing to test it out is our carbohydrate library. So again, we actually collected all of the known carbohydrate antibodies that are out there, many of which are actually in the HIV field. Many of them were actually in complex with antigen with known crystal structures. We used all of that input data, all those antibody sequences to create a library focused on finding antibodies to carbohydrates. And as a result, we created a very diverse library that had different length variation. As you can see on the right, we have quite a bit of kind of shorter CDRs as well as longer CDRs within that population. And there's also some variability in the CDR2 length as well. And then on the light chain, we also can see multiple lengths as well for both CDRs 1 and 3 as well that are, again, probably essential for the behavior and binding of these very unique carbohydrate antibodies. All right, so that was the entire library of libraries. One of the cool things about presenting here today is I actually had a lot of time, so normally I'm not able to actually talk about all of them. So thanks for sticking around to hear about our huge array of different libraries. I'll end with our Tau platform. As I said, we use our amazing library platform coupled with some software that we've created to actually enable us to both humanize and or affinity mature any antibody sequence that people give us. And so this is an amazing platform where we can shuffle natural human diversity that's similar to your particular sequence. And I'll show you in a second how we do that. So the example I love to show is this example where I took a fully human antibody derived from the Cambridge Antibody Technology Library to PD-1. It was actually a triple-digit nanomolar binder to PD-1, but it actually had function and blocked the PD-1, PD-L1 interaction. It was also a lambda antibody. We can, again, put this into the software, and it can basically look and find CDR sequences that are similar within a defined mutational space that you set. So for this particular example, we set a distance of 3. So it gave us all CDR sequences for CDRs 1, 2, and 3 of both heavy and light that were within 3 amino acids of the parent CDR sequence. And we then use that to then build a focused DNA library and put it into phage display to then pan and screen it back against PD-1 to find improved candidates to the PD-1 target. Shown here is an example of some of the affinity data from that campaign. On the bottom right here is the actual parental antibodies. As you can see, it's a fast on, fast off in terms of the overall affinity of that interaction. And as you can see, we found several candidates that had much, much improved off rate, and as a result, lower KD and higher affinity. And shown here is a couple examples of the best candidates that came from the campaign. Again, the parental antibody is here with a 330 nanomolar affinity, and you can see that we actually found multiple candidates that had a single-digit nanomolar monovalent KD. When you actually go in and look at the sequences, you can really see how the sequences are changing in response to driving towards higher affinity. You can see a lot of hot spots in the sequences where particular amino acids are changing in terms of their polar character or hydrophobic character. Another thing is when we build the libraries, we also fold in a lot of additional kind of single amino acid mutations into heavy chain CDR3 as we build it. Initially, it was actually very difficult to find sequences within a defined mutational space for CDR3 because of the huge breadth of sequences in that particular region. As we've actually increased the number of sequences in our databases, we're actually able to make some matches based on similarity. But to add in additional mutants into our library, we also incorporate a single amino acid scan as we build the library. We also find that when we look at these sequences, we can again cluster them based on how homologous they are with each other, see a lot of homology between certain sequences. As a result, we can really focus in on particular sequences that have the highest affinity as well as the best properties. And as you'll see in a second, we also did a full functional analysis of the antibodies. And a really cool take-home message from this particular project is that the affinities we observed actually matched up really well with the functional activity of the antibodies, and that the highest affinity clones actually were the most functional in a blocking assay between PD-1 and PD-L1. So just to summarize, you can bring us an antibody, we put it into our software, it gives us a whole series of sequences that are similar with a defined mutational distance you set. We can use the library team to build an amazing library that we can put into phage display, and then we can pan and screen it against your particular target and hand you back affinity improved candidates. If your original antibody is a mouse or a rat-derived antibody, we can also humanize it as we do that process. Twist is also using our internal libraries for our own discovery and optimization as well to create our own candidates. Shown here is just some of the antibodies that are on the shelf that are available for licensing. We're not a full-fledged drug discovery company quite yet, and so we're looking to license out some of these assets, but someday we actually will potentially take some of these antibodies into the clinic ourselves. So just to conclude, when you think of Twist and Twist Biopharma, these are really all the ways you can work with us. First of all, we love to license out our libraries. So if you're savvy in doing phage display, panning and screening in your own lab, we would happily license the libraries in a subscription-based model. Second, we love to help others generate new antibodies. Of course, we have a whole lab, an awesome team that can basically take in your target and within a 3 to 4 month time frame, deliver back to you fully human optimized antibody candidates. We can also just do an optimization project where you come to us with an existing lead and we can go through that tau process I talked about before. We love to collaborate on new technology as well as new discovery and optimization campaigns around targets that might be of interest to you. And then finally, we're actively trying to partner with others to out-license some of the candidates I talked about before. And so just to conclude, you really should see Twist Biopharma as your partner in antibody discovery and antibody optimization. So if you have any needs in that area, please come and see us at our booth. And that's it. I'd be happy to answer any questions.